So the next talk is by Yingying Wei, right? Thank you. On batch effect correction single-cell RNA. Looking forward to your talk.
Can everyone hear me? Okay. First, I want to thank the organizers for this very precious opportunity. Today, I will talk about valid experimental designs for single-cell RNA sequencing experiments so that we can correct for batch effects. This is joint work with my student Fangda and Angus. So compared to bulk experiments, single-cell experiments allow us to have a much better understanding of cellular heterogeneity. Nevertheless, just like bulk expression data, for single-cell expression data, we can also have very severe batch effects. However, one main difference between bulk gene expression data and single-cell RNA-seq data is whether we know the cell type of each sample. In bulk data, often each sample corresponds to a person, and we know whether the person is a case or control sample, whether the patient comes from cancer subtype 1 or subtype 2. So when the biological heterogeneity is known, so to correct the batch effects, we can apply widely used algorithms such as COMBAT and SVA. However, in single-cell RNA sequencing data, now each sample corresponds to a cell, and the cell type is often unknown and always the target of inference. So then, how can we correct the technical artifacts when the biological heterogeneity is unknown? So in the past several years, there has been very active research in the direction of batch effects correction with unknown sample subtypes. So the RUV normalize try to learn the latent space of the batch effects by assuming we either know a set of control genes whose expression level will not change from condition to condition, or a set of control samples that are repeatedly measured on different batches. However, in single-cell experiments, once we assay a cell, then the cell is gone, so we cannot have replicates across different batches. The metasparse k-means try to tackle the problem from the perspective of joint clustering across different batches. So it conducts the clustering within each batch and try to match the different subtypes across batches. However, for it to work, it requires that all the subtypes must be present on all of the batches, so which may not be feasible for many single-cell experiments. So in the past several years, the MNN and SERET have become very popular. So in MNN, as stated in their paper, it has two key assumptions. So the first assumption is that batch effects variation is almost orthogonal to the biological subspace. And the second assumption is that the batch effect variation is much smaller than the biological effect variation. Well, sometimes the batch effects can be quite severe. That's why we care about batch effects. The recent published Scanorama builds upon the MNN, so it also relies on these two key assumptions. SERET tries to use CCA to learn the shared variations across different batches and treat those shared variations as the shared biological variability. Sometimes, however, technical artifacts can also be shared across batches. Then it can be difficult to distinguish between technical and biological variability. Last year, we proposed a method called BUS that can simultaneously cluster samples across batches and correct for the batch effects. And we applied the framework to analyze gene expression data, microarray gene expression data, to understand the cancer subtypes according to TCGA data. However, all these mentioned methods, although many have already been applied to single-cell RNA sequencing data, they actually do not consider the count data nature of single-cell RNA sequencing data. So we know actually for single-cell RNA sequencing data, we can observe a large number of zeros, which are termed as dropout events. So a large body of literature has tried to model the dropout events. And recently, there are actually also efforts that try to couple the modeling of dropout events and factor analysis. However, the factor models are only approximation to the underlying mixture model. So in summary, when we try to analyze the single-cell RNA sequencing experiments, we are facing severe batch effects, we don't know the cell types, and we have count data which suffer from dropout events, has larger overdispersions, and can also have cell-specific amplification factor due to cell size or sequencing depth. So here we try to tackle all these problems at the same time, built upon our previous framework BUS. So we call our method BUS-seq. So the model is actually quite straightforward. So I'm sorry I cannot, can you see the pointer here? Sorry for that. So for the ICE cell on the B batch, so we assume it belongs to the K cell type according to a probability pi bk. And we allow the cell proportion to vary from batch to batch. Then given the cell belongs to the K cell type, then we assume its underlying expression level follows a negative binomial distribution with a mean of mu Big and an over-dispersion parameters of phi Bg. So we model the log of the mean following the combat format. So it can be decomposed into the contribution from the baseline gene expression level alpha G plus a cell type specific effect. So it models the differential expression between the K cell type and the baseline cell type. And moreover, we let's incorporate the batch effect, which is gene specific. So we let the batch effects to vary from gene to gene. And finally, to account for the cell-specific amplification factor due to sequencing depth or cell size, so we add this term delta BI. However, we cannot always observe this X all the time because we have dropout events. So we use another layer of logistic regression to model the dropout events. So we let the dropout rates depends on the underlying gene expression levels. So finally, suppose there is no dropout events, so then we can have a successful detection, we observe this X. But if we have a dropout event, then we observe a 0 instead. So we are aware some people prefer still to use negative binomial distribution instead of zero-inflated negative binomial distribution to model the single-cell RNA sequencing data. Luckily, so under our model formulation, suppose the data prefers a negative binomial distribution instead of a zero-inflated negative binomial distribution, it can automatically learn it because negative binomial distribution is a special case of our proposed model. So now let's go to the main part of this talk, right? So can we use this new proposed method or any method, right, to always combine two data sets and analyze them together? So let's look at a toy example. So here in the heat map, let's see view each row as a gene and each column as a cell. Then we use different colors to represent different cell types. So suppose on batch 1, we only assay cell type 1 and cell type 2. While on batch 2, we only assay cell type 3 and cell type 4. Okay? So then not only our method, actually no method is applicable because the study design is confounded. So people have long been aware of the danger of a confounded experimental design. So from the very early ages of single-cell RNA sequencing experiments, people have kept on advocating that we should use a completely randomized experimental design. Okay? But unfortunately, everyone is talking, but no one is listening. Okay? So why that's the case? So we can imagine, suppose we want to conduct single-cell RNA sequencing experiments for 10 patients, right? So what a completely randomized experimental design means. It means we need to collect the cells from all these 10 patients, then randomize them to different batches, and send each batch to sequencing. Okay? So this can be a little bit impractical, right, due to all these budget and time limits. So now the question is between these two ends, right? One extreme, we have this completely randomized design that is infeasible, although ideal. And to the other end, we have a wrong confounded design, okay? So whether we can have something between that is valid but still flexible, right? So whether our routine practice, for example, we collect cells from one patient, then we send that as a batch to sequencing. Then come another patient, then we collect all the cells and send that as a batch for sequencing, right? So that different batches, they can share some cell types while they can have their distinct cell types. Whether under such type of experimental designs, we can tease out the technical artifacts from the biological variability, even though we don't know the cell types. So before I go to results, please allow me to quickly introduce the concept of model identifiability. We say a model is identifiable provided that if theta and theta tilde, they give the same probability distribution, then it must be the case that theta equals to theta tilde. However, for a confounded design, then we can find theta that does not equal to theta tilde, but they can give the same probability distribution. So then we will run into trouble. So luckily, we can prove actually under quite many experimental designs. So our model BuzzSeq is identifiable. So first, we prove the simple setting is to say, suppose all the batches contain all the cell types. Okay, so then the BuzzSeq is identifiable. We can separate the batch effects from the cell type effects under mild conditions. So the first condition talks about the odds ratio in the logistic regression for the dropout events. So gamma B1 is negative, means that the highly expressed genes are less likely to suffer from dropout events. So that's actually what we observed in real data. And the second condition is to say, for any two different cell types, there exist at least two differentially expressed genes. So this can also be easily met in reality. So then people will complain, right? Although this one is more flexible than a completely randomized design, in a completely randomized design, we require the cell proportion also to be similar across different batches. So under the complete setting, we can allow the cell proportion to vary from batch to batch, but we cannot guarantee each batch contains all the cell types, right? So then, no worry, so we can prove, actually, suppose you have a large reference batch. In the reference batch, it contains all the cell types. So then for all the other batches, as long as you share two cell types with the reference batch, then we will also be safe. So then people can also complain, right? This can still be too restrictive. How can I guarantee there exists one patient? That patient contains all the cell types collected from all the other patients, right? So then, no worry, we can further prove. So actually, under such type of chain type of design, right? So for every two consecutive batches, like batch 1 and batch 2, as long as they share two shared common cell types, like batch 1 and batch 2, they share the green and the blue cell types, and batch 2 and batch 3, they share the blue and the purple cell types. So then for batch 3, you can have a red cell types and so on and so forth. Then we will be safe. The design is valid. So a special case of the chain type design is suppose all the batches, they share two common cell types. Right? For example, when we collect cells from human tissues, suppose it always contains T cells and B cells for everyone, then we are already fine. So the proof, because it involves the dropout events which correspond to the missing at-random mechanism in statistics, so with development techniques, I will not go through the details, but I still hope that I can explain the intuition a little bit. So why do we need the magic 2, right? So why 1 is not sufficient, okay? So imagine, suppose you observe a Siberian tiger and a rabbit in Russia, okay? So then you go to India. Then you see an animal like this. You see only one animal. Then you get very confused, right? You don't know whether this is a tiger, or maybe a rabbit in India somehow look like this. Or maybe this is another species, right? Okay? But however, suppose you see another animal like this, right? So then all of a sudden, you will know, okay, one is tiger, another is a rabbit. The important idea is to say the contrast between these two species, between two cell types, will keep the same from batch to batch, from one place to another place. So then, suppose you have bear in Russia and a monkey in India, then never mind, okay, so we'll be fine. So then we have shown that actually there are quite flexible experimental designs under which we can correct batch effects. So go back to our model. So we conduct a statistical inference under the Bayesian framework. We propose a parallel MCMC algorithm to conduct a posterior inference, and the computational complexity of the algorithm is linear in the number of cell types and linear in the number of batches. Because this is an MCMC algorithm, so we can automatically impute those X, the expression levels before the dropout events, and also we can simultaneously detect those genes that are differentially expressed between different cell types. We can use BIC to learn the number of cell types, and moreover, like Combat, we can also offer a version of the corrected values. So after you estimate all those parameters, we can remove the batch effects. And we develop a quantile matching approach based on inverse sampling so that the corrected values will keep the biological variability but are absent from batch effects. So we compare our proposed method with the state-of-the-art approaches, including the Liger, the MNN, Scranoma, SCVI, Siret 3.0, and Zimba Wave. So we benchmark the different methods according to the adjusted rand index, which measures the consistency between two clustering results, the Salute coefficient, which measures how separate different clusters are, and also the t-SNE plots. So we analyze the famous integrated pancreas data sets that were first studied in the MNN paper. So the nice feature about this data set is actually we can have some gold standard cell typing for each cell according to the marker genes. So following MNN, so we treat the 4 data sets as 4 batches. So on the left-hand side are the raw data before any normalization. So on the right, then we can see batch effects between the first 2 and the last 2 data sets, quite severe batch effects. So the right-hand side are our corrected values. So after correction, so we can see as if all the data were measured on the same batch. So then we compare the ARIs, and we can see that the BuzzSeq outperforms the other approaches. And also, we can compare the distribution of the salute coefficient, and we can see BuzzSeq also outperforms the other methods. So these are the t-SNE plots, so quite standard. And because we can call the differentially expressed genes, so we conduct pathway analysis on those genes that are differentiate between that differentiate different cell types. So we identified in total 14 enriched KEGG pathways. So among these 14 pathways, 3 are related to diabetes, including the highest ranked one. And the second and the third pathways are related to pancreatic secretion and insulin secretion. And another 2 pathways are related to metabolism. So recall this is a data set that we collected cells from healthy donors and donors with type 2 diabetes. So we think the results are quite encouraging. So in summary, hopefully today I have convinced you implementing a valid experimental design for single-cell RNA sequencing data is actually not too demanding. So actually, if you pay a little bit mind to implement either reference panel design or chain type design, your study will be valid. However, please do keep in mind the magic number 2, 1 is not sufficient. So unless you make very strong assumptions on the structure of the batch effects. So we develop a method that under those flexible and valid experimental designs, so we can craft for batch effects and actually provides one-stop services. So many methods on the market nowadays, it first assume that you will do the normalization. Once you do the normalization, assume your normalization is fine, then they ask you to the imputation. Then suppose your imputation is correct, then they ask you to the batch effects correction. Then assume your batch effects correction is fine, then you do the clustering. Then assuming your clustering is correct, then you do differential expression detection. However, each step can be prone to errors, right? And all the uncertainties are not accounted for. So in contrast, here we propose an integrated model, so it automatically accounts for all the uncertainties together. So currently, our software and also the code for generating all these figures and numbers are available on GitHub. We have an early version available on the bioarchive, and we will soon upload an updated version. So these are the funding support we should acknowledge. And thank you very much for your attention.
Thank you for the talk. We have time for one quick question. Okay, if there's no question, I have a short one. So in the very beginning, you mentioned your batch effects actually depending on the gene, right?
Yeah.
Can you give me a short intuition why this is the modeling choice here?
So actually, I think in gene expression data sets, microarray gene expression data set, and also RNA-seq data, actually we see that quite often. So because different GC content and things related to the transcript can lead to gene-specific batch effects. Yeah.
Right. Any other question?
Sure.
How do you do computationally? Because you do full MCMC. We need to use the microphone. Very quickly, how do you do computationally with full MCMC?
So this is, in terms of MCMC algorithm, is quite standard. But the nice feature about this algorithm is it can be highly scalable. So we actually implement a parallel version for the MCMC algorithm. Yeah.
Okay.
Does that answer your question? Or maybe we can discuss offline.
Thank you. Let's thank the speaker again. We need to move on.
Thank you.